Hello everyone, I'm ManolisKellis and I'll be presenting the work that my lab has been doing on integrative analysis of 10,000 epigenomic maps across 800 samples for regulatory genomics and disease dissection. This is work by Carlos Boix, Ben James, Youngjin Park, Wouter Malleman. So we've basically combined data sets across ENCODE, roadmap epigenomics, genomics of gene regulation. We've used imputation to complete all these data sets, and we also integrated them with genome-wide association studies to basically infer genetic regulation insights and also disease. The basic methodology is that we have uniformly processed 3,000 data sets and imputed another 14,000 data sets, carried out extensive quality control and validation, and then used high-resolution enhancer calls by combining the H3K27 acylation signal across 833 biosamples with enhancer annotations from chromHMM across all biosamples, and also DNase hypersensitive sites across 3.6 million locations in the genome, resulting in 2.1 million active enhancer elements. We've then studied the activity patterns of those elements across the 833 biosamples to infer activity modules and also the relationships between samples. We've also carried out linking between regulators and their target enhancers using regulatory motifs of the upstream regulators, as well as between enhancers and their target genes using a combined approach of machine learning, gene ontology, and correlation of activity. We've used these links and these annotations to prioritize GWAS variants based on the tissues that are enriched genome-wide, the target genes that are inferred from our linking, and also fine mapping based on enhancers that overlap these enriched annotations. And we've also carried out a lot of systems genetics work to understand the relationships between tissues and traits globally across multiple tissues. So our contributions are threefold. Number one, a large data resource that you can find at compile.mit.edu/EpiMap, along with many interactive visualizations of all of our figures. A lot of regulatory genomic insight about specific modules across many different tissues and the regulatory motifs controlling these modules, as well as the linking between enhancers and their target genes. And then lastly, high resolution dissection of genome-wide association studies based on the specific annotations of genome-wide significant SNPs, as well as the enhancers that they overlap and the links that they are, the genes that they are linked to. So overall, the combined resource contains 14,000 data sets across 833 biosamples and 18 different assays. These have been grouped into 33 tissue groups based on both lifestyle, life stage, and tissue type. So these are the 4 projects that were combined. These are the different groups of tissues across the different tissues of the body, the sex of the samples, of the donors, and the type of tissue between tissue, cell line, primary cell, in vitro differentiated cell, as well as the imputation status. You can see that some of the data sets are observed and imputed. Some data sets are observed but not imputed, usually because we did not have any informant data sets. And then other data sets are only imputed, and some data sets are neither observed nor imputed when we did not have enough informants. And then the life stage of each of the samples. So we've separated those into tier 1, tier 2, tier 3, tier 4, based on the density of coverage from observed data. And here's all of the samples laid out end to end. So how do we impute the missing data? We basically use chrome-impute that assumes that no data is known for the target assay in the target cell type, and uses mark-to-mark within sample regression trees and sample-to-sample within mark regression trees, and combines them in an ensemble predictor that basically predicts the target cell type for that mark. Here are some examples of the imputed data in red and the observed data in gray, which was, of course, only provided to the program after the fact to validate just how well the imputation worked. So here's a large number of examples of visualizations showing that the imputed data, in fact, matches the held out data sets very well. This is cross-validation. This is held out data sets that were compared at 25 KB resolution, 200 KB resolution, and 1.5 million nucleotide resolution. And you can see that at all of these resolutions, you capture very well the imputed data, the observed data from the imputed data. We can also compare thousands of random locations across the genome using just a randomization approach, and then seeing how well the imputed data matches the observed data across the genome. So most of the time they match well. In some cases, when the imputed data doesn't match the observed data, we can actually use that to flag low quality data sets. And as you can see, the data sets that have the lowest NSC and RSC strand correlation scores, QC scores from ENCODE, are in fact the ones that are showing the lowest agreement according to our imputation-based QC metrics. We've also used experimental validation to basically show that imputed data matches newly generated data sets that were not available at the time that the predictions were made. And we've also compared the imputed data to the nearest track dataset, as well as the average signal dataset. Now, the nearest track is a particularly stringent benchmark. It basically says, if I were to somehow magically know the best possible predictor track across the entire genome using a single track, how well would I do? And you should note that the nearest track is actually unattainable in practice because it's only knowable after the target track is already generated. So this benchmark is extremely stringent and not even realistic in practice. However, we still outperform it 96% of the time for punctate marks and 77% of the time for broad marks. So Chromium Impute also outperforms average signal 100% of the time for punctate marks and 73% of the time for broad marks. We've also studied the cross-sample relationships using mark-specific clustering, both by stage and tissue. You can basically see here that the combined multi-mark similarity, in fact, groups tissues by life stage in some cases, as well as by type and sex. And this is true for both repressive marks and active marks, which capture different ends of the spectrum. We find that active marks differentiate between different lineages best, whereas repressive marks differentiate between different stages of differentiation best. And you can see here the corresponding projections for each of a number of individual marks. We've used these enhancer annotations to basically study a tree relating all of the samples based on the enhancer activity patterns that they share. And you can see here that this tree, in fact, captures a lot of the known biology of each of those tissues. We've also used Chrome HMM to systematically predict enhancer, promoter, transcribed, repressed, repetitive, and quiescent chromatin states across all 833 reference epigenomes. And overall, the enhancer states, as well as H3K27 methylation and DNAs, this combined high-resolution annotation we've used, covers about 0.8% of each genome, and overall 13% of the genome across all 833 samples. Here's a comparison at scale with the 127 epigenomes of the Roadmap Epigenomics project and the 833 epigenomes of this new collection. So we've built this dictionary of non-coding elements combining these three signals, enabling us to reveal 2 million enhancers and 200,000 promoters, covering a very small fraction of the genome. We've also constructed interactive visualizations across several genome browsers, including the Chrome HMM tracks and this EpiLogos visualization, which is interactive and very, very fast. We've now grouped the enhancers based on their activity patterns across the biosamples, and you can see that about 5% of the enhancers are tissue-wide. They're active very broadly across many tissues, but 97% of them are in fact tissue-specific. So you can see here some examples of enhancers that are active in endothelial cells, stromal cells, blood, brain, embryonic stem cells, and so on and so forth, as well as these broadly expressed enhancers. For each of those modules of enhancers, for each of those 300 modules, we've learned what are the gene ontology terms using GRADE that are enriched in the genes nearby, enabling us to predict something about the function of each of these modules based on the gene ontology annotations of the nearby genes. We've also used regulatory motif enrichments to start predicting the upstream regulators of these enhancer modules that likely lead to their coordinated activity. And what you can see here is a large number of motifs that make a lot of sense for each of those tissues that I'm showing. For some motifs, you basically see very, very broad enrichment, and these very often act in combinatorial ways with additional motifs to define very highly specific processes. Here's some examples, zooming into heart, immune cells, and brain, where you can see very specific biological processes lighting up in those modules and very specific combinations of motifs associated with them. We've also constructed these interactive visualizations, enabling you to explore all of that over at compbio.mit.edu/EpiMap. So you can click on individual enhancers, individual regulators, individual enhancer modules, and then look at the cross enrichments. We've also predicted enhancer gene links using transcriptome epigenome correlation patterns. We've developed a new machine learning approach that does that using feature selection based on a number of benchmark data sets and also incorporating distance. And what we found is that these links are much more tissue specific than enhancers, and they are only found within 3 sample groups and 4 biosamples in the median, while the enhancers at a median distance of 42 kb from the transcription start site. So we've also compared extensively our predictions with a number of other predictions across previous data sets. And what you can see is that for nearly all of the benchmarks, our blue predictions here are in fact performing very, very well and outperforming most of the other methods. We've used that to now predict the cell types of action and the tissues of action for 540 different traits that you can see here across 833 reference epigenomes. We're only showing a very small subset of the traits in order to be able to read the font, but you should already realize that there's a subset of traits that appear to be extremely broadly enriched across many, many tissues. We call these polyfactorial and multifactorial traits, and these are some of the typical traits expected to be very broadly active. By contrast, most of the traits fall only within one sample category. And you can see here this dramatic increase in the number of traits that show enrichment compared to ENCODE 2012 and compared to Roadmap, with now very large number of GWAS studies that show enrichment. That allows us to now start diving into individual examples and basically say, well, these are the enhancer classes that are showing the strongest enrichment, the top rank, second rank, third rank, fourth rank for a particular annotation. In this particular case, for breast cancer annotations from GWAS. We can now see that several of the loci are in fact falling within these top enriched enhancers. And we can now go into these individual loci, look at the GWAS region of association, and in some cases pinpoint a single enhancer where the strongest GWAS signal appears to be localized. In other cases, you see a diffuse set, but what's really interesting is these enhancers are also diffuse instead of highly concentrated as we see here. And these appear to be all linked to the same target gene. So this example is from breast cancer, showing this NTN4 gene. And then this example is from schizophrenia, showing this calcium channel signaling factor, which again makes a lot of sense biologically. We've also looked systematically at the co-enrichment patterns between traits, shown here as individual circles, and tissues, shown here as pie charts, based on the set of tissues that every trait is enriched in. And that allows us to now start classifying traits according to where they're acting. For example, you can see here a lot of brain-associated traits appear to be math ability, educational attainment, intelligence, cognitive performance, but also schizophrenia, neuroticism, depression, and so on and so forth. By contrast, if you look at Alzheimer's, you see that it instead localizes not with the brain samples, but with the immune samples, which makes a lot of sense given the role of microglia in Alzheimer's. You also see in the middle traits such as coronary artery disease that involve many, many different tissues. The beauty of having all of these tissues is that we can now start taking some of these complex polyfactorial and multifactorial traits and dissecting them into their components that they appear to be acting in. So if we take coronary artery disease, we see enrichment in liver, in coronary artery, in thyroid, adipose, and so on and so forth. So we can now start partitioning the genetic variants associated with coronary artery disease into each of these subgroups. And what we found is that the subset of loci that are associated with each annotation are in fact enriched in very distinct biological processes. And if you look at the comorbidity patterns of the corresponding GWAS variants, you see that they're co-enriched, they're comorbid with many processes that are actually quite distinct between the enhancers that are falling within each of the different subsets of tissues. So here's some examples of basically dissecting coronary artery disease between liver and coronary artery. So here's a very liver specific example. Here's a very heart specific example. And here's an example which superficially appears to be liver specific, but then you also see these additional heart specific links, indicating that some of these loci might actually implicate both, both types of tissues for a polyfactorial trait. So here's these examples in higher resolution. We've also developed an interactive browser for EpiMap, enabling you to browse through all of the motif annotations, all of the module enrichments, all of the samples, and select them specifically for download, as well as go through the genome-wide association study results and browse individual linked loci across 30,000 loci that fall within enriched annotations. So overall, we encourage you to explore this new resource that combines ENCODE, Roadmap Epigenomics, and the Genomics of Gene Regulation Consortium. You can visualize them here, and then you can also interact with all of this here. Again, this is joint work by Carlos Boisch, who's leading the study in collaboration with several members of the lab. And we are at MIT and a very friendly bunch. Talk to you soon. Thank you.